recapping your understanding of aerobic cellular respiration. How did it go with your flow charts? Hopefully that was a little bit of a recap. Right. About 10 more folks. Get your votes in. All right. Last call. Get your votes in. One more people. Going once. Going twice. Very last call. All right. You got 90% there. All right. So let's take a look. So the energy investment stage of aerobic cellular respiration, most of you guys are on track, right? That's glycolysis. That's where you're having to start off by investing a couple ATP energy to get the whole thing rolling. It kind of reminds me, if you guys remember that snowboarder analogy that we talked about, when the snowboarder's at the top of the hill, high potential energy, it takes a little bit of energy to activate that motion down the slope, right? The energy to jump off and get started. Same thing with cellular respiration or any chemical reaction. You generally need a little bit of energy invested in order to get it rolling. So glycolysis, that first step of cellular respiration, is always going to require a little bit of ATP energy. So it's considered to be the energy investment stage. So it looks like you guys are confusing it with the Krebs cycle, which is where you take the 2-3 carbon chunks and further break them down into carbon dioxide, releasing all the energy in those bonds. Krebs cycle, remember, is the energy harvesting stage. That's when you're converting the most amount of energy, right? So it's a little bit different. And then recall electron transport chain is when you're building that concentration gradient. It's a way of storing energy in the form of a hydrogen concentration gradient on one side of the plasma membrane inside of the mitochondria. And then chemiosmosis is that last step of opening the ATP synthase enzyme and allowing the hydrogen to rush down their concentration gradient. In other words, allowing them to diffuse as they naturally would. And using that kinetic energy of the ATP synthase turning to cycle ATP. So hopefully you completed that worksheet and you are feeling a little bit more comfortable with cellular respiration, but it looks like you guys have a little bit of work to really nail down those 4 steps. So I would suggest investing some energy. I didn't mean to do that. In studying that process, because it's a big one, right? All right. So photosynthesis is what I'm going to wrap up with. And for photosynthesis, I'm not going to spend as much time as I did with cellular respiration. I want you to have a much better understanding of the overall big picture and how it pertains to climate change rather than learning the nitty-gritty details of all the steps. Cellular respiration, I think, as I mentioned before, is pretty important to how your body functions. And so I like to spend a little bit more time there so that you understand how your body's using energy. All right, so for photosynthesis, we said this is a reaction, catabolic reaction, right, where you're going to be taking reactants and using sunlight energy, converting that sunlight energy to store it as chemical energy in the form of sugar, right? Your byproduct is going to be oxygen. So you're going to be taking the 6 carbon dioxide, popping them together, and pulling the hydrogen off of water to complete the formation of that sugar molecule. And then your byproduct is going to be this O2, right? So this is why photosynthesis yields oxygen, right? Because it's what's left over after you're assembling a sugar molecule. All right. And who's using photosynthesis? We oftentimes will say plants, right? Which is great, right? Plants are really important photosynthetic organisms on Earth, but keep in mind they're not the only photosynthetic organisms on Earth. We have a lot of unicellular eukaryotes that are also photosynthetic. So in this image here, you're looking at a euglenid. It's a unicellular organism that lives in pond water frequently, but also things like diatoms and dinoflagellates that are really important for photosynthesis in the oceans are unicellular eukaryotes. And I think dinoflagellates alone account for about 30% of the photosynthesis on Earth, right? So it's pretty significant, those unicellular organisms. And then down here at the bottom, we're looking at an example of a cyanobacteria. So recall that some bacteria are photosynthetic. The lineage called cyanobacteria is one of the most famous ones. They are the lineage of bacteria that actually invented photosynthesis. So super important. The key difference here for bacteria is recall that they're not using a chloroplast for photosynthesis because they're prokaryotic cells. They don't have a membrane-bound organelle inside, right? So they're just running photosynthesis in a corner of their cell. All right, so big picture, let's think a little bit about how plants work. So here you're just seeing a basic illustration of a generic plant. And we're going to see that plants have shoot systems, what's above ground, and root systems, what's below ground. These two parts are going to be important for acquiring the reactants for photosynthesis. So in the shoot system, this is usually where you're going to see the green aspects of the plant. Remember that most plants are using chlorophyll as their photosynthetic pigment. It is this pigment that's going to be able to absorb sunlight energy and have electrons excited for converting that energy from sunlight energy into chemical energy. So that's going to be in the green tissues, all the little chloroplasts. And the cells or the structure of leaves, I should say, has those specialized guard cells with their stomata on the underside. We talked about these when we looked at tonicity and how plants are using movement of solutes in water to open and close those little stomata where they are exchanging gases. So here's what you can see that's being represented right in here. On the underside of the leaf, you can see this is where the plant is going to be uptaking carbon dioxide. So where we breathe out carbon dioxide, plants are pulling it into their body's tissues. And that carbon dioxide is going to be, again, 6 of them are going to be assembled to make a sugar. And we said the other reactant for photosynthesis is going to be water, right? Water is really important for this process. The place that plant's going to be acquiring water is going to be from its root systems. Here you can see the water and mineral uptake happening. Plants are going to have vascular tissue we talked about earlier in the semester, where they're going to be using capillary action to move that water and deliver it to all of its tissues. It's going to help to maintain structure, but it's also going to be the water that's used in photosynthesis. So again, as you're converting that sunlight energy, you're assembling a bunch of carbon dioxide, 6 to be more precise, together. You've got the carbon, you've got the oxygen. What you still need is the hydrogen, and the hydrogen's going to get pulled off the water molecules. The waste product or the byproduct is going to be oxygen, and you can see that plant is going to be releasing oxygen from its leaves, from those same stomata. You can see the arrow pointing to the oxygen heading out, right? So this is how plants are releasing oxygen. The sugar that's created is going to be transported around the body and used for energy, for running its cellular respiration. Remember that plants have to run cellular respiration in order to be able to survive. Or some of those sugars can be used for storing. If you're maybe a potato plant, right, you might be storing some of that sugar in tubers as part of your root system. And some of those sugars can be used to make cellulose for building new tissues. This makes sense? Big picture photosynthesis. Really important for life, right? So for photosynthesis, our autotrophs, right, are going to be harvesting CO2 and water from the environment, right? And the products of photosynthesis are going to be that glucose and the oxygen. And again, here you can see how the glucose is made up of the 6 carbons and the 6 oxygen that you got from carbon dioxide. And then those 12 hydrogen are going to have to come from water, right? The leftover is going to be that oxygen byproduct. We already said this is going to happen within the chloroplast. Again, you're using chlorophyll A and B, and in lab you got to learn a little bit about the functionality differences in chlorophyll A and B and how they're helping to increase absorption spectrum, the wavelengths of light that plants can absorb. And again, I'm going to just break photosynthesis into 2 pathways, right? So just like cellular respiration, there's over 20 different chemical reactions. We're going to group them into 2 major categories of reactions, what we call the light phase and the dark phase of reactions. The light phase is going to be where you're harvesting sunlight energy, and the dark phase is going to be where you're synthesizing sugars. The dark phase we'll sometimes call light independent phase, which I'll talk a little bit more about in a second. All right, so let's learn nitty-gritties of photosynthetic structures, and my animations got really kind of wacky here. All right, so again, in photosynthetic tissue, we're generally going to be using those chlorophyll A and chlorophyll B pigments, right? That's the primary photosynthetic pigment, although you do see some variations in some organisms that are using different wavelengths of light depending on where they're living. I'm not going to get too much into that. That's more my ecology class. So if we took a look at a plant's leaf, say, and took a closer look, you would be able to see all the cell walls. You can't quite visualize the plasma membrane, but inside you can see all these little nuggets of chloroplasts, right? So this, what you're looking at here in this top animation is here are the cells, and inside they have chloroplasts, and inside the chloroplasts, you can see these stacks upon stacks upon stacks of folded phospholipid membrane. So just like in the mitochondria where those folds, the cristae, allows you more surface area for fitting more electron transport chains, in chloroplasts, you've got all these folds of phospholipid membrane inside so that you can maximize your photosystems. In other words, in a more simplified ways, you can put more sunlight harvesting protein structures that have the pigments with them for harvesting light. And the more of those that you can fit into your chloroplast, the more effective you're going to be at harvesting light, right? And that may change, right, the color of your tissues, right? So we oftentimes see our plants our house plants will sometimes have different colors. Some shade-loving plants tend to have a lot denser number of chloroplast and chlorophyll to be able to harvest sunlight in those shady environments. Whereas plants that are exposed to a lot of light, bright light, don't need as many chloroplasts because they have more access to light. They're a little bit more efficient, so they tend not to be as dark green. But anyhow, plants are kind of cool. Inside the chloroplast, again, you can see all these folds of phospholipid called thylakoids, right? And these little folds of phospholipid, again, designed to increase surface area inside of the chloroplast. Right? The more surface area, the more photosynthesis you can conduct. And then just to give you a visual of the leaves and their stomata. So here we're looking at a rendition of the underside of a plant leaf. And you have these little clusters of specialized guard cells, these cells that are changing their tonicity so that water either moves in or out of the cell. Notice how as water moves into the cell, the gap between the 2 cells opens up. This gap is called the stomata. And this is where gas exchange is going to happen. So we talked about how this is where oxygen is going to leave. This is where carbon dioxide is going to hit in. One of the challenges that plants face is that this is also a space where water is going to be leaving the plant. It's going to be evaporating. So in order for plants to conserve water, they're sometimes going to close their stomata, especially midday, right? When it's really hot and they have the potential for losing a lot of water out of their tissues, they can close their stomata. So stomata are going to help them to regulate gas exchange and water loss. Right? So great little structures, stomata. Okay, doke. So we're going to talk a little bit about the 2 steps of photosynthesis. What are we doing to convert carbon dioxide into sugar, right? How are we harvesting sunlight energy? Again, there's 2 major pathways within photosynthesis. So this is what we're going to talk about next, right? The light phase and the dark reactions, or the light phase or the light reactions. Those terms are used interchangeably. Remember that we talked last week about oxidation reduction reactions. One of our reactants is going to be getting oxidized, one's going to be getting reduced. Again, we're not going to go into as much detail, but remember that the photosynthetic process shares a lot of the characteristics with cellular respiration, primarily electron transport chain, and chemiosmosis is going to be used during photosynthesis. We're just not going to talk about it in as much detail as we did for cellular respiration. So here are the 2 main steps that you need to know. The light reactions, what you need to know about them is that this is where you're harvesting sunlight energy, right? And if you're harvesting sunlight energy, that means you require light for these light reactions. This is why they're called the light reactions. They can only happen during the daytime when there's sunlight availability. So light reactions need sunlight energy. And we're going to see that the light reactions are going to harvest energy and temporarily store them, right, in NADPH. I'll talk about this in a second. And that energy that's stored can then be used to synthesize sugars. And this is what's going to happen during the dark reactions. This is the synthesis phase. This is where you're producing sugars, where you're assembling carbon dioxide back into a sugar molecule. The dark reactions historically were named maybe not so accurately, right? By calling them the dark reactions, it gave people the impression that they only happen at night, and that's not the case. The dark reactions can happen during the day, or they can happen at night. The significance of the dark reactions being separated out is that now it allows plants to harvest sunlight during the day. And if it's a hot day, you can harvest sunlight and keep your stomata closed, right? And then at night, when it cools off and you are less risk of losing your water, then you can separate out your dark reactions and use the energy you harvested during the day to synthesize sugars. Right? We now know that dark reactions can happen during the day as well, right? So to call it the dark reaction is not so popular anymore. We oftentimes will see it now called the light independent reactions, meaning that they don't require light to happen. Does that make sense? Probably more than you needed to know. All right, so what's happening during light reactions? Again, this is where you're harvesting sunlight energy. That sunlight energy is going to be exciting electrons in the photosynthesis systems, the aggregations of protein and chlorophyll that are inside of the thylakoid membrane. Right? And the excitation of those electrons, again, is how we're going to be able to convert energy. We're going to use that kinetic energy of the moving electron and convert it into a temporary chemical energy. So for cellular respiration, we talked about NADH, this temporary energy storage molecule. We're going to see a very similar molecule that's going to be used to temporarily store the energy that you harvest during the light reactions. It's called, and you don't need to worry about this big old name, but here's the fancy name for it. It's called NADPH, right? So it has an extra little P in there, and it always reminds me of pH for like photosynthesis, right? So this is the temporary energy storage molecule that's used by photosynthesis. A very similar process to what we saw in cellular respiration. That NADPH is going to be fueling the electron transport chain. It's going to be how you're going to cycle ATP in order to run your dark reactions. So the NADPH will be produced or cycle during the light reactions, we're going to store it and then use it later on in our dark reactions or the Calvin cycle. With me so far? Right. So this is where we're taking sunlight energy. This is also when we're breaking down water, right? It's called splitting water. I'm not going to talk too much about that, but it turns out that this in the light reactions is where the oxygen is being produced. And then in the dark reactions, you're just assembling the sugar. All right. So again, light reactions, you're going to be breaking water apart. You're going to be harvesting sunlight energy. This is happening in the thylakoid membrane inside of the chloroplast. You're using that sunlight energy to excite electrons, and you're going to store that energy that's harvested as NADPH and some ATP. That harvested energy can now be used, right, to run the dark reactions. Okay, so we've got the energy so far. We harvested it during light reactions. Can only happen during the day. Now let's talk about the dark reactions. Right? The dark reactions are sometimes called the Calvin cycle or the light independent reactions. Right? So they have a few different names. I generally call them light independent, but I'll probably use dark reactions on the exam since that's what I used as a header here. So for the light independent reactions, remember that they can happen during the day or at night, right? They just don't require sunlight energy, right? So they're independent of light. They're going to actually be occurring in the space inside of the chloroplast, so outside of the thylakoid membrane series of stacks. And what's going to happen during the dark reactions is that we're going to take carbon dioxide, 6 of them, and we're going to use that energy that we harvested during the light reactions to form bonds between the carbon. So those 6 carbons will be attached back together. 6 carbon dioxides will be attached back together. And this is also where we're going to take that hydrogen that was pulled off of water and use it to contribute to building that sugar. Right? And at the end of the dark reactions, we're going to end up with sugar that can then be used for cell functions. Right? So you have to have both light and dark reactions. Light reactions can only happen during the day. This is where you're harvesting the energy. The dark reactions can happen during the day or during the night. And this is where you're assembling or synthesizing sugar from carbon dioxide. Does this make sense? Light and dark reactions. I think at this level, photosynthesis and cellular respiration, or photosynthesis is a little bit more easier to understand than cellular respiration, but in part just because we're not going into as much of the detail. Any questions? Photosynthesis? Nothing? Okay, all right. So I did want to spend just a minute or two talking about photosynthesis and how it applies to climate change, because I think it's a little bit more important to understand about photosynthesis and cellular respiration and how they apply to everyday life, right? And as we are seeing more and more impacts of climate change, I think understanding how photosynthesis and cellular respiration connect to that are important. And I'll talk about climate change in more detail later on in the semester. But carbon dioxide, remember, we said we're generating when we run aerobic cellular respiration, and every time we exhale out, we're releasing it into the atmosphere. And this is true for all living organisms, right? So cows are producing CO2, plants are producing CO2, bacteria are producing CO2, fish are producing CO2 and releasing it into the atmosphere, right? And this is where it's going to accumulate. And then the way that we bring it back into the terrestrial system is by photosynthesis, whether it's oceanic plankton or oceanic plants or land plants or unicellular eukaryotes. They're going to be all inhaling that carbon dioxide and fixing it back into sugars that can be used to fuel the food web, right? And in fact, photosynthesis is the basis of a food web. We call it primary productivity. It's really important, right? And so I think that it's important to understand that photosynthesis is a really important mitigator, potential mitigator for climate change, right? Because plants, as you learned, I don't know, did you guys play the carbon cycle game as part of the lab? for photosynthesis. Oh, no, I think it's an ecology lab. Never mind. So you'll learn a little bit about carbon cycle later on in the semester when we start talking about ecosystems. But this cycling of carbon dioxide, pulling it out of the atmosphere, is a really important aspect of photosynthesis. It's one of the only ways that carbon can be brought back from the atmosphere into the food web. And what we're seeing now with climate change is that we're adding extra carbon dioxide into the atmosphere by extracting fossil fuels, which again are dead bodies of organisms, and burning them, right? Land conversion's also a big contributor to climate change. As we're chopping down trees and vegetation, we're removing the photosynthetic capabilities of Earth's biosphere, right? And so as a consequence, we're disrupting the cycle of carbon dioxide that normally happens, right? The carbon dioxide that's being released by cellular respiration has always been balanced, right, with the carbon dioxide that's getting pulled out of the atmosphere for photosynthesis. And so photosynthesis is oftentimes, used as a potential mitigation for climate change. I don't know if this is going to run. There it goes. So here's a cool little animation from Cal Academy of Sciences looking at photosynthesis rates in the land ecosystems and then the ocean ecosystems across the span of the year, right? And notice how it changes over the span of the year. What is happening here? Why are photosynthetic rates dropping? I'll play that one more time. Any ideas?
Because of the seasons?
Because of the seasons, right? And so here you can see winter receding, right? So in spring, your photosynthetic rates are going to increase, right? Notice how most of your land mass is in North America. Here's the equator here, right? And then here comes fall and winter again, and photosynthetic rates are going to drop, right? You get seasonality in photosynthetic rates, and that's going to have impacts on carbon dioxide levels. Oops, right, which is what you see here.
Hi, this is Bill Putman. I'm a climate scientist at NASA's Goddard Space Flight Center. What you're looking at is a supercomputer model of carbon dioxide levels in the Earth's atmosphere. The visualization compresses 1 year of data into a few minutes. Carbon dioxide is the most important greenhouse gas affected by human activity. About half of the carbon dioxide emitted from fossil fuel combustion remains in the atmosphere, while the other half is absorbed by natural land and ocean reservoirs. In the Northern Hemisphere, we see the highest concentrations are focused around major emission sources over North America, Europe, and Asia. Notice how the gas doesn't stay in one place. The dispersion of carbon dioxide is controlled by the large-scale weather patterns within the global circulation. During spring and summer in the Northern Hemisphere, plants absorb a substantial amount of carbon dioxide through photosynthesis, thus removing some of the gas from the atmosphere. We see this change in the model as the red and purple colors start to fade. Meanwhile, in the Southern Hemisphere, we see the release of another pollutant, carbon monoxide. This is a gas that's both harmful to the environment and to humans. During the summer months, plumes of carbon monoxide stream from fires in Africa, South America, and Australia, contributing to high concentrations in the atmosphere. Notice how these emissions are also transported by winds to other parts of the world. As summer transitions to fall and plant photosynthesis decreases, carbon dioxide begins to accumulate in the atmosphere. Although this change is expected, we're seeing higher concentrations of carbon dioxide accumulate in the atmosphere each year. This is contributing to the long-term trend of rising global temperatures. The Orbiting Carbon Observatory 2, or OCO2, will be the first NASA satellite mission to provide a global view of carbon dioxide. OCO2 observations and atmospheric models like GEOS-5 will work closely together to better understand both human emissions and natural fluxes of carbon dioxide. This will help guide climate models toward more reliable predictions of future conditions across the globe.
Anyhow, so you can see the impact of photosynthesis on being able to mitigate carbon dioxide levels. Now, while we really need to reduce our emissions, right, that has to change. This is why we see this big push of restoring vegetation, right? Planting trees, restoring grasslands, types of vegetation that grow relatively quickly and can pull a bunch of carbon dioxide down. Keep in mind that right, we've removed quite a bit of vegetation on the Earth's surface, and if we can restore a significant amount of that, or maintain the integrity, I should say, and maintain the integrity of the vegetation that we have now, we can really help to impact climate change, right? This is especially true of the burning of the rainforests in the tropics. Right? It's a really important carbon dioxide processor at the global scale because less seasonality there. You've got more consistent photosynthesis over the span of the year. So the burning and destroying of the rainforest that's happening in Brazil right now is really significant for global climate and the future of climate change. Yeah. Anyhow, any questions on cellular respiration or photosynthesis? Doing okay? Nothing? Are you ready to learn about cell division? Ooh. All right, so I'm going to go ahead and start cellular reproduction. Oh, let me toss a poll. A little bit worse at polls this week. I didn't put a photosynthesis poll in there. All right. I'll do one a little bit later. Sorry. Gardeners are here. Maybe some barking. We'll see. All right, so we're going to move on to talk about cellular reproduction, or how cells reproduce. Throughout the course of the semester, we're going to be talking about reproduction at multiple scales. So we're going to talk about reproduction at the cellular scale right now. And remember that this is important because many of the organisms that are on Earth are unicellular, and this is how they're reproducing. Later on, we'll talk about how multicellular organisms reproduce when we talk about reproductive systems and focus on human reproduction. So reproduction, this term, means the creation of new individuals, and it can happen at multiple scales, right? If you're a unicellular organism, you're going to be able to use mitosis for reproducing and creating new unicellular organisms, but it is also how we reproduce cells in our multicellular bodies, right? So we're going to talk about cell reproduction and focus in on how cells divide, right? And so you might have noticed my subtle differentiation between cell reproduction and cell division. They are not interchangeable terms, right? Cell reproduction is the replication of the cell and the cell's contents, right? So replicating the organelles, replicating the DNA, right? That happens all during a stage called interphase that I'm going to talk about in a minute, versus the divvying up of that replicated material, which is what we call cell division. So cell division is the divvying up of the material. It's independent of the replication process, right? So we'll talk a little bit more about that in a bit, but first let me, hey, it's okay, come on. Let me talk about the types of reproduction on Earth, just to kind of give you some context. So we're going to talk a little bit about this today, and then I'll talk a lot more about asexual versus sexual reproduction when we get to reproductive systems next unit. So asexual reproduction, remember, I think I've pointed out this word root before. Means the absence of. So in asexual reproduction, you have no exchange of genetic material, right? You're just replicating yourself. And indeed, some organisms can use asexual reproduction for making a genetic clone of themselves. So for example, we'll see sea anemones, right? Aggregating anemones can make copies of themselves by ripping their bodies in half and then replicating and preparing the damage that's been created by that process. And at the end, you'll have two sea anemones. Right? For asexual reproduction like this, whether we're talking about binary fission or budding or the different types of asexual reproduction, are all going to be using mitosis exclusively, right? Bacteria are going to be unique. I'm going to talk about bacteria for just a couple minutes today and talk about how they divide their cells. Remember that bacteria cells are less complex than eukaryotic cells, so they're going to use a less complex and different mechanism for replicating themselves. Bacteria are going to use a process called binary fission, and I'll talk about that in a minute. For sexual reproduction, this is where you're exchanging genetic material with another individual. And what that means is that you're going to have to take your full set of DNA, your chromosomes come in pairs, and segregate those pairs apart so that you provide your offspring one half of your genetic material and your partner provides one half of their genetic material. In order to do that and shuffle your genes together to make an offspring, you're going to need a special type of cell called a gamete, right? It is a cell that has half of your genetic information. It is a sex cell. We talked about this earlier in the semester. And to make those cells, we use a special process called meiosis, right? So I'm going to talk about mitosis, and I'll probably have time to get to meiosis, but we'll see. I might have to finish up meiosis on Thursday. So we're going to talk about the two major forms that eukaryotic cells use for dividing. Remember that prokaryotic cells are using binary fission. They don't use mitosis. And binary fission, right, is slightly different, right? Again, less complex mechanisms. In a prokaryotic cell, they're going to take their bacterial DNA and copy it, right, replicate it, and then they're just going to pinch off and divide the cytoplasm with the content so that you end up with two cells, right, at the end. Genetic copies. Right? Binary fission, again, can, because it's a less complex process, can happen quicker than eukaryotic cell mitosis. So we can see, so in the animation here, you can see how bacteria can divide at a fairly fast rate and have quite a bit of a population explosion. So some bacteria can actually replicate every 20 minutes, right, which is pretty remarkable for a generation time. So one of the things that helps us to understand how bacterial infections, for example, can explode out of nowhere is they have the potential to replicate themselves fairly rapidly. Again, binary fission is a type of reproduction that bacteria are using. It's not sexual, it's not genetic exchange of material, and because they're prokaryotic cells, it can happen at a faster rate. You guys hear me okay over that lawnmower? Seems particularly loud today. I think it's just because I don't have the AC on. All right, so we're gonna focus on cell division and to talk a little bit about when it happens, when do we use cell division, and what are the steps of cell division. And this is what you're gonna be looking at in lab, is how do we identify the different stages. So I'll talk about that a little bit as we go through as well. We mentioned cell division earlier, where we said all cells are going to come from a pre-existing cell. We have to copy a pre-existing cell set of genetic information in order to make a new cell. So this is what we're going to talk about today, is how do we copy pre-existing cells. For replication and cell division by mitosis, we're going to see that this process is going to be important for a variety of functions, right? When you are growing. So when you're growing from a fertilized egg to embryo, or from an infant to a toddler, you are adding more cells to your body by cell division, right? So growth, right? Anytime you're growing, as your hair is growing, as your skin is replacing itself as you abrade it every 2 weeks, right? You're using cell division, you're using mitosis. Whenever you damage your tissue, as in the little GIF here, you can see that you repair a cut, say, or any injury by replacing the cells and making a little cell patch, right? Mitosis, mitosis, mitosis for that. Maintenance, right? Your cells, for the most part, have a lifespan. That lifespan can vary according to the type of cell that you have in your body. And there's a few cells that don't exhibit mitosis, like your skeletal muscle cells, but most all your cells in your body are replicating themselves and replacing themselves over time. Even your bone cells, right, replace themselves in their entirety about once every 7 years. Right? So you're constantly breaking down bone and adding new bone. So maintenance of your cells. And keep in mind that cell division isn't going to be synchronous. Not all the cells in your body are going to, boom, divide all at the same time, right? Different cells in different places are going to be dividing at different rates, right? So again, your skin cells are going to have a really fast rate of mitosis. So the skins in your epidermis, for example, are replacing themselves every 2 weeks, right? Your stomach lining cells, right, that are experiencing that acid pH that we talked about, replace themselves every 2 days, right? So some cells that have a lot of abrasion or wear are replacing themselves relatively quickly, whereas other cells that don't have as much of abrasion, like your skeletal system cells, replace themselves at a much slower rate, pardon me. Right? And as I mentioned, some cells don't replace themselves by mitosis at all. So your skeletal muscle cells have only limited ability to repair themselves. All right. Any questions on the function of mitosis?
Really quick.
Yeah.
Can you just really quick define mitosis again, please?
Oh, I'm going to talk about that in just a second.
Okay.
Yeah. I should mention, one of the things that's not on here is asexual reproduction. Anytime an organism is reproducing asexually, they're going to be using mitosis. All right. So we're going to talk next about the cell cycle. And I think that this is an important concept because when we spend a whole lecture talking about mitosis and meiosis, students walk away thinking your cells are dividing all the time, right? And while they are dividing all the time, it's not the only thing that cells are doing, right? And in fact, they're dividing usually only a small fraction of their lifespan, right? So we're going to talk about this thing called the cell cycle, right? Which is essentially an understanding of the lifespan or the life cycle of a cell, right? What happens at different stages of the cell's functionality, right? And so we can see from this diagram and from the text here that cell cycle can be broken down into 2 major phases that happen during the cell's lifespan. Interphase, which is when the cell is doing its normal cell jobs, right? So if you're a skin cell, you're doing skin cell jobs. If you're a salivary gland cell, you're doing salivary gland cell jobs, right? Producing enzymes or whatnot, right? This is also where cells are going to be growing, right? This is also where cells are going to be replicating their DNA and their organelles. A lot's happening during interphase. And one of the things that we'll learn is that in your average cell, interphase accounts for about 90% of the cell's lifespan, right? Or 90% of the cell cycle is spent in interphase. We think it's all spent in mitosis, but mitosis is actually just a small part of the lifespan of a cell, right? And again, some cells never undergo mitosis, like your skeletal muscle cells. All right, so interphase, normal cell jobs, and I'll talk about this a tiny bit more in a minute, versus what's called the mitotic phase, right? And that's what's illustrated here in blue and purple, right? The mitotic phase. And notice that the mitotic phase is broken down into 2 steps, mitosis and cytokinesis, right? And so what I want to point out here is that mitosis and cytokinesis are 2 different steps, 2 different stages of the division process, right? And if you notice, they're both encompassed in this mitotic phase, right? So the mitotic phase and mitosis, another way to say this, they're not interchangeable terms, right? And oftentimes students will walk away thinking that mitotic phase is the same as mitosis. It's not. Mitosis is just part of the mitotic phase. You also have to include cytokinesis. And we're going to talk about what's mitosis and what are the steps of cytokinesis so that you understand them pretty well. And hopefully by tonight, you'll be muttering the steps of mitosis in your sleep. All right. So one of the other things that you might notice is that interphase has been broken down into subphases. And I'm not going to talk too much about the different subphases of interphase, but I do want to note that there are certain spots that are considered to be checkpoints, right? And this is really important in cell regulation. So before a cell divides, right, or after a cell has divided and is growing, right, the cell has a moment of what I call cell reflection, where the cell self-assesses, right? Am I mutated? Have I been infected by a virus? Is there something wrong in my cell? I might want to consider not replicating, right, if I'm infected, right? Or if I have a cancerous mutation potentially in my cell, your cell has the ability to halt replication. Because remember, when you replicate an infected or a mutated cell, the two daughter cells that result are also going to be infected and mutated. And you don't want that in your body. You want the ability to, kind of keep that infection or disease in check. So these checkpoints are really important, right? For regulating that process. And then also notice that there's a checkpoint during mitosis so you can assess if anything went wrong. Does this make sense? You have genes that are helping to regulate this process to help you to, give cells basically the permission to divide when they have no mutations or to halt, right? And depending on the types of genes that you have, you might be better or less able to detect precancerous cells in your body. So if you are self-reflecting as a cell and detect that there's a mutation in your cell or an infection, your cells can undergo apoptosis. So not only do they not divide, but they can undergo apoptosis, which is defined as programmed cell death. They can self-destruct. They kill themselves so that they don't perpetuate that infection or mutation, right? So apoptosis is a really important way to protect the body against disease of multiple types, right? I do want to mention that apoptosis is also really important in growth and development, right? So what the images here are showing you is how apoptosis is used during embryonic development. Right? We have to go from a ball of cells to a three-dimensional organism. And that's going to involve adding cells to some places, but killing off cells in other places. So here you can see an embryonic human hand developing, right? And over that process, you're going to see an enhanced rate of mitosis at the tips of each of these lobes. And then you're going to progressively see apoptosis happen in between to separate out the digits. So this happens when you're making fingers or when you're making toes or when you're making legs or when you're separating arms off of the torso. Apoptosis is really important for development. And in the middle picture here, you're looking at a genetic condition called syndactyly, where there's errors in messaging for apoptosis during development. And what you can see is that individuals with syndactyly have fused digits, either toes or fingers, where apoptosis failed to happen during development. Right? We also have genetic conditions called, I think the common name is mermaid syndrome, right? Where sometimes during development, the legs don't separate out. There's no apoptosis. And so the babies are born with their legs fused together still, right? So apoptosis is a really important process inside of our bodies, right? So we're going to talk about cell division a lot, but remember that cell destruction is a really important aspect of how your bodies are functioning. Good so far? I lost track of my time. All right. What we're going to do in the next few minutes is talk about these major steps or stages in the cell cycle. We're going to talk a little bit about interphase, what happens during interphase, again, 90% of the cell's lifespan. We're going to talk about the steps of mitosis, how we segregate contents of the cell and begin to divvy them up. And then cytokinesis, the final stage is where we separate out the cytoplasm and result in two cells, right? Two duplicate copies of cells, right? So first step we're going to talk about is interphase. In interphase, again, this is where most of your cells are going to be spending most of their lives doing normal cell jobs, right? So growing, producing enzymes, or protecting against dehydration, depends on where your cells is. If you're in the liver, maybe you're breaking down toxins, right? So cells are going to have all kinds of different functions, right? This is also the stage in which at the end of interphase or before the cell enters into mitosis, the cells are going to be replicating DNA, right? And one of the things that we're going to learn is that DNA, in order to be replicated or to be accessible for normal functions, we're going to have our DNA in the chromatin form. So this is what we're going to talk about next, is what are some of the different forms of DNA and why do we change the form of DNA at different stages, right? So again, during interphase, we're going to be duplicating the DNA, and that's basically where you're taking advantage of the molecular structure of that nucleic acid. You're going to be separating apart the two halves of the DNA strand, exposing the nitrogenous bases of your nucleotides. And don't worry, we're going to come back and talk about this next lecture in more detail. And you're basically creating two strands, right? So you separate one strand and complementary base pair match it back up so that you have two strands that were made from the original strand. And again, I'll come back and talk about this in more detail when we get to DNA structure and function next class. So here in this top illustration, you can see how the DNA strand is unraveled during interphase, then complementary base pair match up so that original strand is preserved in the two new strands that are generated, right? So you can see the pink half strand of the original strand, and then the purple is what's been added in. So every time your cells divide, that cell's divvying up its genetic material and giving half of each DNA strand that's been replicated. And coincidentally, this video here this animation is real time, right? So this is about the rate at which your DNA is replicating. So that's kind of cool. All right, so what are the different forms of DNA? So I'm going to go through some terminology before I stop talking about the steps of mitosis. So we're going to talk about chromatin, sister chromatids, chromosomes, and daughter cells. Right? So let's talk about what these terms mean. And for the first three, it's looking at what's the state of the genetic material, right? And so this top picture here is the chromatin form of DNA. So you can see that you've got all the strands accessible and unraveled. All these strands are going to be changing form when you're getting ready to divide. Those same strands will be highly condensed into what are called chromosomes, right? So chromatin and chromosomes are two forms of DNA, right? On the same strands, but in different states, right? So chromatin, right, is unraveled DNA. And most of the time in your cells, your DNA is in the form of chromatin. And most people don't realize this because all of our pictures, all of our visualization of DNA is when it's in the condensed form. It's when it's most visible for us, right? But most of the time, your DNA is in the form of chromatin, right? Which is that double helix loosely wrapped around some proteins called histones. And what is significant about this chromatin stage is that the DNA double helix strand is accessible. It's accessible so that you can separate the strands apart and replicate them. it's also accessible so that you can read and implement the instructions that are stored on that DNA strand. And again, we'll talk about gene expression next lecture. When I imagine DNA, I imagine chromatin in your cells as a big mass of spaghetti where all those DNA strands are unraveled and all intermixed and tangled with each other. Right? Now, here's the key thing. For cell division, each one of those DNA spaghetti strands is going to have to be replicated, right? And then sorted so that you make sure that only one of each of those DNA strands is in each of the daughter cells, the two cells that result, right? That would be like untangling and separating out this big mass of spaghetti, right? And not making any sorting mistakes and not having any of the strands tangle up on each other and damage the DNA. Sound like a fun job? No, right? And it would cause all kinds of problems, right? So what your cells are going to do is they're going to condense the DNA, right? They're going to change chromatin into chromosomes, right? So here you can see what chromatin looks like, loosely wrapped around histone proteins. And here you can see the beginning stages of how we condense the DNA, right? So here we're looking at a nucleosome. It kind of looks like a little strand of beads. And then we're going to slowly begin to wind up the DNA. And then my other animations didn't work for some reason, and condense it even more. And then loop each of those condensed strands and then condense them even further into a chromosome, right? So here you can see the nucleosome, these little beads, right, of DNA wrapped around proteins called histones. They start to coil around and they loop up and then the loops coil up, right? And so that you get this highly condensed structure called a chromosome, right? Now, notice the DNA is all embedded in there, no longer accessible. The formation of a chromosome is like the equivalent of taking a spaghetti strand and condensing it into a bow tie pasta. Right? This is only going to happen when the cell is getting ready to divide. Right? And it's really important, right? Because now, instead of having to separate out a nasty bowl of duplicated spaghetti, now you separate out bow tie pasta. Is it easier to sort? Is it easier to pull apart? You betcha, right? So the formation of chromosomes are an organizational step that happen during a division. Again, because those DNA strands are all condensed, it makes it really easy for us to take a picture and visualize DNA. So most of the images we see are of chromosomes. But keep in mind, the chromosome only forms temporarily when the cell is dividing. And we'll talk about those stages in a bit. All right. So chromosomes are going to exist in the nucleus, our genetic material. Again, most of the time in the chromatin form, but as we're getting ready to divide, they're going to condense up into these strands called chromosomes. And I'll talk about, what happened to my sister chromatid slide? PowerPoint gremlins. There it is. Okay. I moved it. All right. So the chromosome is, keep in mind the condensed package. Every organism on Earth is going to have a different number of DNA strands of chromosomes. They're going to vary in number, right? More doesn't necessarily mean better, right? For a human, we've got 23 pairs, right? A total of 46 chromosomes. And again, note that your chromosomes come in pairs. I'll talk about those pairs in a little bit. The pairs are going to vary according to the species, right? So dogs have 39 pairs. This genus of butterflies here has 134 pairs. That's crazy. Fruit flies have 4 pairs, right? So very little DNA. This is one of the reasons why we use fruit flies so frequently in genetic studies, is because it's been easy to sequence and identify all the genes in fruit flies. There's not that much genetic material there. Again, your chromosome is a package up DNA strand that is a double helix of nucleotides that's condensed up with proteins called histones to make this discrete packet. And again, only going to be in the chromosome phase during the mitotic phase when the cell is going to be dividing. And then last characteristic I want to note is that your chromosomes are going to come in pairs. Those pairs are called homologous pairs. And homologous is a term that indicates shared ancestry. And in the case of your homologous chromosomes, what's happened here is that if we're looking at this sample chromosome, notice how one's illustrated in red and one's in blue. And what it's trying to convey there with those color differences is that you inherited one of each of your chromosomes from each of your parents, right? So for your pairs, you inherited one from your mother and one from your father, right? So for each pair of chromosomes, you have one from each parent. And they're homologous because what they share is the sequence of genes, right? So here you can see just a really generic gene A, gene B, gene C. Homologous chromosomes share the same gene sequence. So if gene A is the gene for noses, it's always going to be found on this pair of homologous chromosomes. And it means that you have two genes for how to build a nose, one that you inherited from mom and one from dad. Right? How you express those genes we'll talk about later when we get to inheritance next week. Right? Makes sense? Homologous pairs share the same gene sequence. And every one of your pairs, 1 through 23, if you're talking about humans, have a different sequence of genes. Each pair of chromosomes has a different set of genes on it. All right. So when you are dividing your cells, remember that you replicate your DNA first during interphase. And in the first steps of mitosis, you're going to start to condense up that DNA into chromosomes. And you're going to take a second organizational step in forming what are called sister chromatids. Remember that you duplicated this chromosome or this DNA strand, right? So you're going to take the identical copies, right, and attach them together at this little neck here called the centromere, right? And that's going to help you to keep the two identical copies paired together so that when you go to divide, you can say, oh, these two are identical. I'm going to put one in this cell, one in this cell. Right? So it's a way of organizing the duplicate copies of DNA, right? So they are identical strands. Copied the strand during interphase. When you condense it up, you're going to pair it up with its identical copy. And remember, during that division process, you take an original strand and divide it in half and complementary base pair match it up. So each one of these chromosomes has half of the original strand and one half new strand. But I can talk more about that for DNA. Good with a sister chromatid, identical copies of chromosomes. When they're bound together at the centromere, they are considered to be one chromosome, or they're counted as one chromosome. During the cell division process, you're going to be pulling apart the sister chromatids and distributing one of each of those chromatids to the new developing cells. Right? The two new cells that are produced during cell division are called daughter cells. They are identical copies of each other in mitosis. Somebody, oh, there it is. Okay. Sorry, I was feeling like I didn't know what time it was. All right, so we're gonna dive into mitosis next. Again, remember your cell spending most of its time in the green here in the interphase, right? This is where the DNA was already duplicated. Your organelles would have duplicated at some point during interphase as well. And what we're going to talk about is the mitotic phase, specifically mitosis, right? And I'm going to walk you through the 1, 2, 3, 4 steps of mitosis. We're going to learn what happens during each step and how to identify each stage of mitosis, right? So I'll show you some pictures, right? So again, mitotic phase is made up of mitosis and cytokinesis. And we're going to see that the mitotic phase or mitosis right, that happens during the mitotic phase, is going to be broken down into these 4 steps: prophase, metaphase, anaphase, telophase, as if they were distinct 4 chunks of things that happen. But keep in mind that mitosis, right, is actually a process. It's like a dance, right? So what you're seeing here in this middle animation is the process of mitosis happening. And notice how it doesn't pop between the 4 stages. It's transition. So the 4 stages represent 4 snapshots of this dance called cell division. So the early stages are going to involve the formation of chromosomes and sister chromatids. They're going to get pulled around, aligned, and then separated out, right? So again, they're going to get organized, aligned, pulled apart, and segregated out. Right? We're going to walk through each one of these generic stages. Keep in mind there's intermediate stages, and you're going to learn all about that in lab as you're trying to identify what's prophase versus metaphase. Right? In order to move those chromosomes around, you can't do it by magic. You have a whole series of protein fibers that are going to be acting as a pulley system to separate those chromosomes around, to organize them and separate them. This series of proteins is called the mitotic spindle. So remember that animal cells have this centriole, and the centrioles during early stages of mitosis are going to migrate to opposite ends of the cell, and protein fibers are going to grow out of that centriole to make this structure called the mitotic spindle. It's going to be growing from either end of the cell, and a protein strand from one end is going to attach to a pair of sister chromatids, and a protein strand from the opposite end is going to attach to each sister chromatid bundle. And that's then going to act like a pulley system to move those chromosomes around and then eventually pull the sister chromatids apart. Yeah? Okay. All right. So mitotic spindle, critical for this process to work. So again, for each of the stages of mitosis, I'm going to walk you through what happens during each stage. You should know the stages. You should know their order. You should know what happens during each of the stages. And then I'm going to tell you a little bit about how to identify the stages just to help you out for lab, right? So repetition. So during prophase, this is the first step of mitosis. During prophase, this is where your chromatin is going to start to condense and form sister chromatids. Chromosomes and sister chromatids. So when we take a look at a cell, right, all these cells, most all these cells are in interphase, right? And I can tell it's in interphase because I can see a discrete round nucleus, and inside, right, I don't see any strands of DNA. I just kind of see this murky, shady area, right? That's because my DNA is in the chromatin form still, right? It's not condensed enough that I can visualize it as a strand, right? In interphase, you can also, if you squint, see the darker spot inside of the nucleus. That's the nucleolus, right? That area where ribosomes are going to be being made, right? So in early prophase, what you're going to start to see is, it's going to start to look like strands as those chromosomes start to condense up, right? So you're going to be converting chromatin to chromosomes, and as you do so, look at here, you can see the nucleosomes, right? And later on, you'll be able to see the actual sister chromatids, right? As this progresses, right, the nucleolus is going to magically disappear, right? And the nuclear envelope is going to break apart because remember your mitotic spindle outside in the cytoplasm, your DNA in the nucleus. In order for the protein fibers to attach to the DNA strands, you're going to have to break apart the nuclear membrane, the structure that's isolating and keeping those two things apart from each other. And as you can notice kind of the subtleties in prophase. See how, nice discrete round looking nucleus. Notice how here it's looking a little less spherical, and even less here. Right? What you're actually seeing is as the nuclear membrane starts to disassemble, the genetic material starts to spread out, right? You get less of a discrete looking nucleus. So the spindle fibers, notice how the centrioles are migrating, the spindle fibers are forming. Right here you can see the shady purple nuclear membrane break apart. Sister chromatids come together, right? And by the end of prophase, right, all of your sister chromatids are going to have mitotic spindle attached to it. And notice how spindle is going to come from either end and attach to those sister chromatids at their centromere, that little waist part of the chromosome, the invaginated part. Good? So prophase. If we look at it under polarized light, right, here's our cell. This green represents the centrioles. You can see all the strands of protein fiber growing out. Notice how this is much later in prophase. You can actually see DNA strands or chromosomes in this case. And the spindle's starting to grow and attach to all of those chromosomes. They're just starting to come together to form sister chromatids. Maybe you can spy that in this little setup here. All right. What happens as it progresses is that spindle starts to pull those chromosomes around, right? And aligns them in the center midline of the cell. Organizational step. This is the step known as metaphase, where your sister chromatids are all aligned up at the center plane of your cell, right? And what you're doing is you're kind of organizing them so that in a couple minutes, what we're going to see is that spindle fiber, once it arranges all the cells, all the chromosomes in a midline, it's going to start to pull from opposite ends until it rips apart those sister chromatids. So it kind of reminds me of lining up during recess, right? So you're nice and organized, right? And then you can separate out readily one sister chromatid to each of your subsequent daughter cells, right? So it makes sure that you get one of each copy of that DNA strand into each of the two subsequent cells that are going to be forming. So metaphase is when they're lined up, right? And then the spindle fibers are going to be pulling from opposite directions. And as soon as they pull apart, we're going to see our next stage. So again, here's polarized light. Depending on how many chromosomes in the organism, we're going to see either more linear midline aligning up, or it can sometimes look just like a mess of chromosomes. But you can see that they're clearly clustered in the midline of the cell. And notice how the spindle fibers, right, look about equal size, and that's because, right, they are, as they have aligned up, their width represents aligning up at the midline of the cell there. Okay, so the spindles are pulling from opposite ends, and the instant that those sister chromatids pull apart, you are now in anaphase. And then as anaphase progresses, the spindle fibers start to retract to opposite ends of the poles. And in that process, they're going to pull one of each sister chromatid to either end of the pole, what's going to be designated as the two new cells that are going to be forming, right? So here you can see anaphase where there's two distinct clusters of genetic material in there. It's a sudden action, right? You can oftentimes identify anaphase because notice as the spindle fiber pulls at the chromosome's little arms start to wave in this V-like fashion as it's getting tugged around. And you'll oftentimes see that V-like shape to the chromosomes as you're visualizing cells. Right? So for example, in here. Make sense, anaphase? As anaphase progresses and the cells start to get pulled, or the chromosomes start to get pulled to opposite poles of the cell's also going to start to elongate a little bit. And so if we take a look at our, polarized light dividing cell, notice how there's two distinct clusters of DNA strands here. And see how the spindles are getting shorter as they're pulling the two clusters of chromosomes to opposite sides of the cell. Now you want to elongate and pull your chromosomes to opposite ends so that you can clearly designate what will become one of each of your new cells, right? So our next step is telophase, right, where your cells are getting even more and more elongated. Your sister chromatids are getting isolated to opposite ends of the developing two new cells, right, to opposite poles. And then as telophase progresses, right, see the white line appears, right? That's the beginning of cytokinesis. So telophase and cytokinesis overlap a little bit, right? So what's happening during telophase? You're getting ready to start to reassemble the two daughter cells that are going to be produced by this process. And so telophase is oftentimes described as the opposite of prophase, because everything that happened during prophase is going to get reversed during telophase. So your chromosomes, right, are now going to start to unravel back into chromatin. Your nuclear membrane is going to reassemble to re-isolate that DNA inside. Your nucleolus that disappeared during prophase is going to reappear. Your mitotic spindle that formed is going to break down, right? So everything that got progressed during prophase will get reversed during telophase. The nice thing is now that you've learned those 4 things that happen during prophase, right, you can now know the things that also happen during telophase. You just have to add elongation and the beginning of cytokinesis happening. Right? And then if we take a look at the plant tissue over here, you can see how you've got 2 distinct little clusters of genetic material, and see how you can't see the strands anymore, right? It's kind of starting to look like a big mass again. That's because the DNA is starting to unravel. You're not going to be able to see the distinct strands as that chromatin starts to reappear. And again, here's our polarized light. Same principle, right? Now look at the blue genetic material. See how it's getting more shady instead of being able to see distinct DNA strands? That's because the chromosomes are unraveling back into chromatin. Your mitotic spindle is breaking down. Your nuclear membrane is going to refold. Your nucleolus is going to reappear, right? You're putting those 2 cells together. So here you can see they're still sharing cytoplasm. So what we're going to do last is cytokinesis. So mitotic phase, remember, is made up of 2 stages. Mitosis, which is prophase, metaphase, anaphase, telophase. And then the second process that happens is cytokinesis. And it is defined as the division of the cytoplasm. So this is where you're going to be divvying up the organelles and the cytosol, right? And that's going to happen by using, again, protein fibers that are going to literally pinch that cell apart so that the plasma membrane all comes together and forms 2 daughter cells. So at the end of cytokinesis, you're going to have 2 genetically identical daughter cells produced, right? Plants, animals, single-celled eukaryotes are all going to use this process. The big difference that happens between plant and animal cells is that in 2 daughter plant cells, you now have to separate them by the formation of the new cellulose cell wall, right? So you get the cell plate that forms during cytokinesis, and that step does not happen during animal division. Yes? All right. So probably should have done so. Hey, knock it off. Let's see. How about delinquent on polls today? Got a little poll there, recapping, recapping the functions of mitosis. So you should be able to, so what you're going to do in lab this week is practice identifying mitosis. Keep in mind that sometimes you're going to see intermediate steps of mitosis. So there's intermediate steps, and you just have to make your best call at what you think is being observed, right? So here's a nice metaphase. Here's a anaphase. Here's a telophase, 2 distinct isolated bunches. Here's an interphase, here's a prophase, right? DNA starting to condense. Metaphase aligned up. Anaphase, you can see the V's. Telophase, notice how the chromosomes are disappearing as the chromosomes are unraveling. All right. So get familiar with those steps of mitosis. All right, last call for poll. We've got 25 out of 37. And then, you can practice quizzing yourself as to what's happening during the different stages. And so you should know the stages of mitotic phase, mitosis and cytokinesis. For mitosis, you should know the stages, what happens during each stage. Last call for poll before I move on and talk about some applications of mitosis. All right, so let's take a look. We are doing pretty good with our understanding of mitosis. So the question was, which of the following is not a function of mitosis, right? So remember that growth, maintenance, repair, and asexual reproduction are the functions of mitosis. You're adding new cells to grow, you're replacing worn cells for maintenance, you're repairing damage to tissues by adding new cells, and then if you're reproducing asexually, you're making new cells to make a genetic clone of yourself, right? So the one step that is not part of mitosis is apoptosis. Remember that apoptosis is that programmed cell death. This is what we use to destroy damaged, mutated, or infected cells. So just go back and check your progress on that, because it looks about half of you guys are on track. And then there's a little bit of confusion out there. So go back and recap. All right, so I probably should have talked about this earlier. We talked about that cell control system, which is the cell checks where the cell can identify damage, mutation, and undergo apoptosis. Right? What happens if you don't identify a precancerous cell and you allow it to replicate? This is what cancer is. Cancer is a mutation that affects the cell cycle control system, where it disrupts the cell's ability to undergo apoptosis and prevent it from dividing. There is usually some type of mutation that causes malfunction of the cells as well. Right? For cancer, we're going to see lots of different types of cancer out there. And the types of cancer are going to depend on the type of cell that was afflicted. Remember that you have over 200 different categories of cells that make up your body. Each type of cancer is going to be afflicting different types of cells. So, for example, carcinomas are cancers that are referring to cancers that are on the outside of your body. So all types of skin cancer. But within skin cancer, we have different types of skin cell cancer. We've got basal cell carcinomas, we've got melanomas, and that again is a reflection of the different types of cells that are in your skin. Depending on whether a melanocyte gets infected or a germinative cell gets infected, you're going to have different types of skin cancer. This is why cancer is so challenging to solve as a human disease, is because when we talk about cancer, we as general population think, oh, all cancer is the same, and it's not, right? They all use different mechanisms, and there's different lineages of cancers that are interacting with our cells in different ways, right? So there's no one cure for cancer. All right, so what happens if a potentially precancerous cell has its cell cycle control system disrupted? It can't self-check in apoptosis, and so it's going to divide by mitosis. And once it divides, you now have two cells that have that mutation. And those two cells divide and you have 4 cells. And those 4 cells divide and you have 8 cells, right? And eventually this will develop into what we call a tumor. It's a cluster of abnormal cells that have developed from one cell that was the problem child at the beginning, right? And then depending on how that tumor interacts with neighboring cells, for some cancers, the cancer cells can secrete a chemical message to the neighboring cells that causes them to have their cell cycle disrupted as well, right? So this is what's called a malignant tumor, right? Where it can communicate to neighboring cells and affect them. Compared to a benign tumor, which is a cluster of cells that are just the descendants of that original damaged cell. They're isolated. They're not being transported to other places in your body. So for a benign tumor, this is a good kind of cancer, right? Because the cancer cells are all in that one cluster in that location, right? And so a surgeon can go in and remove the tumor and remove all the mutated cells, and then it's gone. But keep in mind, if you miss even one cell's left behind, right, then that cell's going to divide by mitosis and make 2. And those 2 cells are going to make 4. And those 4 cells are going to make 8, right? And that tumor is going to come back. Right? This is why when you have a tumor removed, right, your doctors are always talking about the margins, right? And the strategy is to go in and remove the tumor, but to also remove healthy tissue around it, right? So that you're sure that you got all the cancer cells, right? All the damaged cells. Because again, all you need is one for that tumor to come right back. So the problem with this is that sometimes those tumors are attached to really important nerves or really important blood vessels, and you can't completely remove them all safely, right? So benign tumors versus metastatic tumors. And this is a type of cancer where in the tumor, that cluster of cells, you can slough off cells, and that one cell can now be transported by either your lymphatic system or your bloodstream to another location, and it's going to start to divide and make a tumor there. And another cell can maybe slough off and head to another part of your body and form a tumor there. So metastasis is when you now have tumors all throughout your body from these sloughed off cells. Much harder to treat, because even if you go and remove all of these clusters, right, you still don't know about the individual cells that are floating around and still haven't resulted in a tumor, right? Much, much harder to treat. Does this make sense? Cancer? A little bit application of what happens when mitosis goes awry. All right, I'm gonna stop there, and next class I will finish up with meiosis. So I would like for you guys to review and be really familiar with the stages of mitosis, because we're going to build on that in order to understand meiosis. Let me throw a last pull out your way. So you should know each of the 4 steps and what happens during each of the steps. And then we'll talk about how those steps are going to also be used for meiosis with some twists, right? So if you understand the steps of mitosis well, then meiosis is going to make a lot more sense for you. So you've got about a couple days to kind of get familiar with mitosis. If you have lab early in the week, that'll hopefully help you to review. And then for meiosis, we'll come back and talk about why it's important for cellular reproduction, sexual reproduction, pardon me, not cellular reproduction.